Hello, everyone. Welcome to our event tonight. Thank you for being here for Berkeley Arts and Letters. This is our event for Dr. Euan Angus Ashley, the launch event for his new book, The Genome Odyssey, Medical Mysteries and the Incredible Quest to Solve Them. We're honored to be hosting the launch event tonight and are grateful to Steven Quake, a co-president of the Chan Zuckerberg BioHub, for joining Dr. Ashley in conversation tonight. My name is Evan Karp. I'll be your host for the evening. I'm the events manager for Booksmith. We're an independent bookstore and mainstay of San Francisco's Haight-Ashbury district since 1976. This event is part of our Berkeley Arts and Letters series, which features exceptional authors and provocative thinkers with new books. Just a little bit about the book. Since the Human Genome Project was completed in 2003, the price of genome sequencing has dropped at a staggering rate. For reference, it's as if the price of a Ferrari went from $350,000 to a mere 40 cents. Through breakthroughs made by Dr. Ashley's team at Stanford and other dedicated groups around the world, analyzing the human genome has decreased from a heroic multi-billion-dollar effort to a single clinical test costing less than $1,000. For the first time, we have within our grasp the ability to predict our genetic future, to diagnose and prevent disease before it begins, and to decode what it really means to be human. Here to discuss the book with Dr. Ashley is Steven Quake. He's the Lee Otterson Professor of Bioengineering and Professor of Applied Physics at Stanford University and is co-president of the Chan Zuckerberg BioHub. He received a BS in physics and MS in mathematics from Stanford University in 1991 and a doctorate in theoretical physics from the University of Oxford in 1994. Quake has invented many measurement tools for biology, including new DNA sequencing technologies that have enabled rapid analysis of the human genome and microfluidic automation that allows scientists to efficiently isolate individual cells and decipher their genetic code. Quake is also well known for inventing new diagnostic tools, including the first non-invasive prenatal test for Down syndrome and other aneuploidies. His test is rapidly replacing risky invasive approaches such as amniocentesis, and millions of women each year now benefit from this approach. His innovations have helped to radically accelerate the pace of biology and have made medicine safer by replacing invasive biopsies with simple blood tests. And Euan Ashley is a professor of medicine and genetics at Stanford University. He was born in Scotland and graduated from the University of Glasgow. He attended Oxford University, completing a PhD there before moving to Stanford University, where he trained in cardiology. He joined the Stanford faculty, where he led the team that carried out the first medical interpretation of a human genome. Ashley has received awards from the National Institutes of Health and the American Heart Association. He was recognized by the Obama White House and received the Medal of Honor from the American Heart Association. His articles have appeared in many journals, including Lancet, the New England Journal of Medicine, the Journal of the American Medical Association, Nature, and Cell. He appears regularly on local and national radio and TV. He's the founder of 3 companies and advisor to several Silicon Valley companies. With 3 young children, he spends his spare time trying to understand American football, play the saxophone, and conduct research on the health benefits of single malt Scotch whiskey. This last area of interest we definitely share. And it's an honor to have you both here. Thank you. Euan, congratulations on your book. And Steven, thank you again so much for joining us. I gladly turn it over to you too.
Great. Thanks so much, Evan. It's really wonderful to be here and to be part of such an amazing legacy and of dedication to books and to publishing. And Steve, thanks for joining us. Thanks for being part of the launch today.
Well, it's a pleasure, and I'm so happy to see your book get launched and to share your story with everybody.
Yeah, so before we start, you got to tell me about that background that you have there, because it looks a little bit familiar, but...
Yes, that's the top of Mount Ellis overlooking Lake Tahoe on Valentine's Day. And that's my son in the orange and our guide, Sam Hamilton, in the red as we were skiing in the backcountry there. And you were standing right behind me when I took the picture, of course.
I was, and fortunately, we have managed to see each other in person, which has been great. And outdoor activities, in particular skiing, have been good for that. So although I've been missing many of my friends and have not managed to see very many close family friends and relatives over the last year, we have managed to spend some time together. Of course, there was a time when we didn't know each other, and so that's where the book starts. And I thought it'd be really fun to just jump back in time and talk a little bit about that, because where I start the book was a moment where I winded my way around the incredibly confusing Clark building at Stanford. And maybe at one point someone will explain to me how the numbering system on that building works. But I had to find my way to your office. But But tell me a little bit first before we get into where the book starts, just the part before that, because I describe in the book how the Human Genome Project cost $3 billion. It was 10 years, 10 countries. It was a gargantuan effort. But then over the course of the years up to, say, 2008, the price of genome sequencing dropped, as Evan mentioned, like the price of the Ferrari down to 40 cents from like $400,000. That was kind of amazing. But you contributed to that. So why don't we start there? Tell us about your genome, and then we'll go from there.
Yeah, I had a very small role in all that. And, it was a very interesting time. I'd say in the late 90s, the genetics community kind of decided they should do this giant moonshot project and try to sequence the human genome. And it was just unimaginable to do at that point with the technologies in hand. And a lot of folks got interested in that as a problem outside of biology. So myself as a physicist kind of crossed into my world. There's a need for new technology to do sequencing that allow us to solve the human genome. And so that got my interest going in trying to develop new sequencing technologies. It became part of my research as a professor. While I and many other people are trying to develop new sequencing technologies, sort of what happened is the powers that be decided to go ahead and just brute force the whole project, which might cost $1 billion using kind of the best technologies at the time rather than waiting for new ones. And so the first human genome came out with this outrageously high price tag, but right on its heels, there were many, many people developing new technologies, all of which helped lower the cost. And I was part of that group. And the sequencer I developed, the single molecule sequencer, provided a factor of 10 decrease in genome sequencing costs at that point, where it went from, there were another technology, it was sort of $500,000. We took it down to $40,000. It was a big step. And reduce the labor. It was like the first time you could sequence the human genome without needing a room full of sequencers and a huge team of people. Just one lab could do it. And so that was a significant advance. And we decided to do my genome first, in part because I was curious about it. In part because in those early days, it was just, really hard to imagine how you would sort of ethically consent someone to sequencing their genome and releasing all that data there. I figured, I'm in a better position than most, and I'm willing to carry the risk. So we did that at Stanford in my lab. It was Norman Neff and Dmitry Pushkarov and myself. There were just three of us, and all I did was kind of sit around. Norman did all the sequencing, and Dmitry did all the computation associated with it.
You gave some blood, though, I think, didn't you?
I gave some blood, exactly. Yeah. Yes. Yeah. And that was a big deal for me because, I tend to faint around blood and all that. So they were teasing me for being brave. But, once we had it sequenced and assembled, the question is, what do you do with it? And there was nobody had done that before. There wasn't really a way to do it at the time. And so that was a very frontier area. And as you recount in the book, that day you came to my office, I was trying to look at my genome using this very primitive tool developed by George Church at Harvard and his students. And, they were trying to make the first genome annotator. They called it the Genomatic. And I was kind of playing around with it when you walked in for our meeting.
Yeah, and I remember that well. I described this at the beginning because I thought that, and I say this in the book, that your office is not, I don't think you'll mind me saying, the tidiest of places. There are many journal articles piled high. It seems in my mind that, to the ceiling of your office. And there you were, and I knew of you by reputation. I don't think we'd even met before. We might have been in the same room at a conference, but and there you were, kind of in the middle, at this keyboard, like staring at the screen. And I know we were supposed to be talking about something else, but we ended up talking mostly about your genome, and that was partly because you showed me the name of a gene on the screen that I recognized. And I remember scrolling through, here's my genome, and to me that was just, my jaw was on the floor, because at the time we were used to sending genetic testing that took 3 months to come back and cost $5,000 or $10,000, and we'd get 3 genes, the sequencing to spell out the sequence of 3 genes. And I think there had only been 4 or 5 people who in the world at that point who'd sequenced their genome. So for me, as someone who practices genetic medicine, as a cardiologist at Stanford, this was mind-blowing, and my jaw was on the floor. But I picked it up off the floor in order to pay attention to the variants that were scrolling by, and you showed me one in in this gene myosin binding protein C that I knew of as the most common cause of a condition called hypertrophic cardiomyopathy. And so that's a condition that comes up a few times actually in the book. But the first time it comes up is in this early chapter when you were showing me your genome. So at that point, our conversation changed a little bit, and it became a little bit more doctor-patient. And I don't know, do you remember what you were telling? Your family basically had been telling you to go find a cardiologist, I think. Tell me about that.
Yeah, I mean, for sure. We had, as you kind of describe in the book, we had a history of heart disease in the family, but it's not one that anyone wants to think about when it's in your own family. You can easily kind of discount those things. It's not really going to affect me and so forth. But this is why the doctors who are objective are very helpful. And my father had some heart problems, and my cousin's son had passed away of unexplained heart failure. And you kind of put the pieces together and said, Steve, you got to think carefully about this, and maybe there's some answers in your genome that'll help us understand how worried you should be about this and what you should do, and how you should take care of yourself.
Yeah, and I remember that distinctly. You're saying your family had been sort of pushing you to find a cardiologist, and I just happened to kind of wander in, and here we were suddenly talking about your genome. So I remember inviting you to my clinic, and we pulled some favors and got you some testing. Do you remember that testing? Do you remember what you did?
Oh, I do. I was very proud. You told me I had the cardiovascular system of an athlete half my age. I felt very good about that.
Right. Yeah.
That didn't make the book, unfortunately.
Right. No.
That particular comment, I love that.
Yeah. But your fitness number did, though. I seem to recall describing that things were reassuring, and that was a pretty good number. So yeah, we made you sweat. Your doctors are fun friends to have. We put you on treadmills or in bike in your case, and make you go as hard as you can. Everything's very safe and you're monitored carefully, but we make you sweat and go to max. And thankfully, we didn't find anything to worry about directly in imaging your heart. It was more when we turned to some of your blood tests and your lab tests that we started to think that maybe we had found something that could help us, give us a clue, because once we turn to the genome, we need some clues for where to look. And your cholesterol wasn't quite, should we say, not quite perfect.
It wasn't great. That's right. And I had never heard of Lp(a) until we started talking about it. That was a whole new sort of topic for me to understand about. It had to do with blood chemistry and heart disease.
Right. So yeah, the folks who are listening might be familiar with good cholesterol and bad cholesterol. That's sometimes called LDL and HDL, low density lipoprotein. And lipoprotein is just a way of moving fat around the body, and it comes in different densities. But you had a high level of a more exotic lipoprotein called Lp(a) as opposed to capital A. It's a small a, and that's a particularly strong version of these lipoproteins that can cause atherosclerosis or hardening of the arteries. And so yeah, we started to feel that this might help explain some of what we'd seen in your family. And so then we turned to the genome, but that was a new thing, because at that point, Dimitri in your lab, I think, had been building some tools, which was great to just allow us to call these variants. But then what do you do with all those variants? How do you amass all the information that's known about genes and health in order to build it into a tool that could be used for medicine? And so that was when we started to call a few friends. And Ross Altman was one of the first, Atul Butte, who's now up at UCSF. Another group were one of the first two I called. And then we talked about your choice of sequencing yourself. And so it was very clear, both of us were very clear that if we were going to do this, we should really engage our ethics team at Stanford and Hank, really world-renowned medical ethicist. And that became our kind of core team at the beginning. Well, but tell me, what were you thinking at this point? Okay, so you knocked it out of the park on the bike, so maybe a little bit of reassurance. But you had literally just walked into a clinic with your genome, and to our knowledge, no one else had ever done that before. This is an unusual situation.
Yeah, and it was, a really seminal moment, I think, in the history of medicine, where a patient walks into a doctor's office and says, "Here's my genome, treat me," and you guys totally rose to the challenge. It was really spectacular. And just reflecting on how we did that, it was also very unusual, because not only did I have my genome, I was the one who had sequenced it. And so I knew, kind of what the how much confidence to have in the sequence in various parts. Right. And so I ended up being in the room as you guys were deciding how to do the clinical annotation. And as you mentioned, we had the ethicists involved, and Kelly Orman, who was the genetic counselor. You mentioned her in the book. Yeah. I just have this very vivid memory of us, all being in the conference room and going over some of the results and having a bit of an argument about whether you could trust this or that. And I'm in the middle of the argument, and she's just there with her head in her hands, because that's not the way it's supposed to work.
Yeah.
Right. The patient is supposed to, not know about these things happening behind the closed door. But I was in this unusual position that I felt I knew which parts of the annotation we could trust or not.
Yeah, really unusual situation for a patient. It was not, we used to call you patient zero, but we were particularly unusual because you were, your team and you specifically were part of the team that had generated the data and that were then part of working out what we should do with it. And we're not normally used to trying to create new tools and thinking what's real and what's not, and where the error might be with the person whose genome it is, in this case, the person who would be affected and whose family would be affected right in front of us. But it actually in many ways laid the foundation for the next few years and the next few genomes we were going to do, where in many cases, the people involved were actually part of the analytical team, which was very, very unusual. And I know that then at the time, we had, in the conference room, we were basically just trying to invent a way to do this. What does it look like? Can we think of tools to visualize a whole genome? I often say this because we talked about cholesterol, we're used to like 5 numbers. Like when you go to your doctor, they do your cholesterol, they get 5 numbers back. Suddenly we're dealing with 6 billion letters. And medicine just isn't set up to deal with 6 billion letters. And even at the time, our computers and our algorithms were not really set up to do that. And some great groups before the team at Baylor, there's a team in Seattle had really worked on a few different ways of thinking of genetic variants, not just for single genes, but more broadly. And we very much stand on their shoulders. But this, I think, was really the first time we'd had a whole team thinking of what is the maximum amount of information we can get from this one genome. And first of all, we wanted to make sure we ruled out the variant I'd seen on the screen that day in your office. And thankfully, Matt Wheeler, who led a lot of this analysis in my lab, was able to rule that out and reassure us. So we saw that your heart was looking good. You had the heart of an athlete. Was it an athlete or a horse? I can't remember. It was one of those two.
I remember athlete.
All right,
half my age. I remember that too.
Half your, yeah. I can see why. So that was good. But then also we had to move on to the fact that your cholesterol numbers were not quite where they should be, and were there clues in your genome for that? And actually, we did indeed find, you mentioned LP little a, there's a gene called LPA, 3 capital letters, that is responsible. And it's an unusual gene because sometimes your genome can expand from generation to generation, and it's a gene that has expansions in it. And normally expansions in genes are bad. They certainly don't sound like they would be good. LP little a is a funny protein, and LPA, the gene, is funny because as it gets bigger, actually your level of LP little a gets smaller and it's actually safer. So unfortunately, yours was a bit too small, and so your LP little a level was high. And we certainly were wondering about that. And we're then in this interesting situation where we had that genetic information. We had to decide, do we recommend a cholesterol medication? And so, at the time, there was this complicated spreadsheet, a sort of tree of to try and help doctors decide. And we plugged in all your information at the end, and it basically said, use your clinical judgment. It was like, complete, it wasn't helpful at all. So we were back to like, well, what do we do? Do we need to look more broadly and use whatever information we have? And in your case, we had a whole genome. So we thought somewhere in there must be a way to help us decide whether we should push you to take a statin because of your risk or not. And that's where the pharmacogenomics came in. And I think afterwards, you'd said that was one of the more important things, I think, that you thought came from the project.
Oh, yeah, absolutely. So Russ Altman's group, who had done that part with you, and they're sort of world experts in pharmacogenomics that built the biggest database, gave me a list of drugs. And they said, these will work with you. And these, you should worry about side effects. And if you ever get prescribed, this is sort of what you should look at. And that felt like a very practical thing to have in your back pocket if you're ever, in the hospital or whatever, and very actionable and a very concrete sort of outcome of this sort of genome analysis. And on that list were statins. You're a responder to these statins, right?
Yeah.
Will work
Yeah.
for you, and you won't have side effects.
Yeah. And that was kind of an amazing thing for us too, because really, although we understood and knew about Russ's work, his whole team came to the table for this, which was amazing. That was the case with each of the teams. And they built new tools essentially to try and apply this to your genome. And in this case, we were interested to see both that you had a variant that would suggest you'd respond to the statin, and that you had another variant that would suggest you were less likely to have the side effects. So I remember then sitting down again with Matt Wheeler. I'm just looking at that photo, remembering he was on top of that mount as well. So this was one of the things. Yeah. Basically, none of us knew each other that well before this project, and we had all become really good friends for the last 10 years, which has really been amazing. So Matt was there, and the two of us as cardiologists were sitting in my office going, wait, we now have a patient, we have a genome, we have the best analysis we could put together with just the best people we knew to help. And at the end of the day, it came down to this decision. We had to make a recommendation. And on the one hand, that was really simple. Every day a doctor sits in an office, primary care doctor, cardiologist, all around the world, and makes a decision whether to say, take a statin or not. But we were really aware at the time that we didn't think or knew of any other doctor anywhere in the world that had a patient's whole genome to help them make that recommendation. And so the weight of that we definitely felt. And in the end, of course, as you recall, we definitely recommended that you took the statin. So
what did your patients say after all the in-depth analysis?
Yes, well, I was going to come to that, because we're due to revisit this because if you recall, we published the paper, and we were very grateful to have a great reception, and lots of people said very kind things about it. But we actually did an interview together. In fact, it might be the last time you and I were sort of recorded on camera was with NPR, and specifically they did a doctor-patient interview, and they asked you specifically, after I said I had recommended the statin, they asked you if you had taken it.
And I said no. I'm very stubborn. I felt like a very young person, too young to be on medicine, for sure. And I had the number you needed to treat. I had a big argument about the number you needed to treat to avoid one event, and it just didn't seem right to me. And just I haven't told you this part of the story, Euan. It might not surprise you. I come from a long line of very stubborn people, and
that does not surprise me at all.
My mother, who turned 80 last month, 2 years ago, her doctor said, you should be on a statin. And she said no.
And she said no. Yeah.
Eventually they got her to do it, but she hated the idea in her late 70s of having to be on regular medicine. Just did not like that. Didn't make her feel healthy, and she resisted all the way. So you should be very proud that you eventually got me on the statin before my 70s, and early in my late 40s, you got me to do it. And it took years for you to wear me down. You're a great doctor and very persistent, I'll tell you that.
Well, that is very kind of you. And I had a little bit of help. Josh Knowles, who's one of the cardiologists at our
you guys double teamed.
We did. Yeah, I realized that you probably needed to hear it from a few more people. So even with the weight of a genome, you still needed two doctors to push you over the edge.
Well, the argument that was convincing in the end to me was that it's not going to do much for you right now, but it's an investment for when you're in your 70s. And in that decade, presumably you make it to your 70s, it'll have your chance, cut in half your chance of an event. And I figured, all right, that seems like a good investment to me.
Yeah. Well, I'm glad that you did. And there are a few medicines for which there is this level of data on the benefit in primary prevention, which is what we're doing with you. So, well, I'm glad that we finally completed that circle and had a chance to talk about that, because neither of us knew he was going to ask us that on the air. So I think we both knew the answer, but I wasn't sure that it was going to come up. So that was fun.
Yeah, it was.
I wanted to spend a minute talking about your cousin. I know that you mentioned Rich and his son Richie, and we describe in the book, just what an incredible kid he was, and the fact that one morning he didn't wake up and he died suddenly as a teenager. And we see a lot of families in our clinic for whom that happens, and it's just it's not even possible to comprehend how hard that is. But obviously this happened to your family, and I know Rich was close and he reached out to you. Tell me a little bit about that.
Yeah, it was absolutely devastating, right? I mean, to lose a child at such a young age, and whole family was distraught, and I try to put myself in the position of Rich and his wife, and they want to know. I'm first of all looking for closure. I mean, there's no explanation; it just happened, and there's a desire for a sense of could we have done something? What caused this? And then we've got a daughter too, and should we be doing something for her? Is she at risk for anything? And so anything similar. And so they were reaching out to everyone they knew for thoughts and help. And I got a call from Rich one day, and he knew I'd sequenced my genome, and because it had been in the news. And so I said, well, you should talk to Dr. Ashley and see if he can help you at all. And you were wonderful. I mean, absolutely. You had them come out, and actually we went and sequenced Richie's genome and did an enormous amount of detective work to try to figure out what might have caused his death. And in the book, you talk about all this and about sort of some of the false starts and the wrong paths and sorting things out, and so I think you conclude with what your best guess is. And many of these things, it's hard to be definitive, but at least you have some information and a best guess for what it is. And I think that was very helpful for the survivors. Yeah,
and it's been amazing to get to know Rich and the family, and just to connect, obviously. And one of the joys that we have in the work that we do, even though at times it's touched by family tragedy, is that we get to know families and we see their fortitude through events like this. But the power of the genome, I think, is really to try and help those who are left behind. And one of the amazing opportunities that we had in this situation was to apply genome sequencing really for the first time to the tissue of somebody who died. So someone who was no longer with us, to try and help those who were left behind. As you said, there were a number of twists and turns, and this starts to, the story in the book starts to talk a little bit about how we actually do the analysis and how we understand. We talk about the fact that often it's not trying to find a needle in a haystack, but rather trying to find a needle among a whole bunch of other needles. It's like a needle in a stack of needles, because everybody has genetic variants in the genome. And now with the inventions that you've made and others, we have power to spell out the genome. But the next challenge is then trying to work out what it really means. And although we definitely hope that there was a provisional answer there, we've continued to work on that on Richie's genome over the years. And in fact, in the most recent time, this is just in the last few months, we have started working with the teams who have been sequencing ancient DNA. And so again, the folks who are listening might have heard just last week, there was, I think, a mammoth. Was it a mammoth? I think there was sequence that was over 1 million years old. So you hear of these humans, Neanderthals, animals like mammoths and others that are perhaps frozen after death in the permafrost. And so those investigators have developed these exquisitely sensitive tools for dealing with fragile DNA that's been, 1 million years is just an unbelievable time. And so, but they have techniques of really maintaining the DNA in a way that we didn't have available to us 10 years ago. That field has really jumped forward. So most recently, we've been working still with one of Richie's samples to try and use those techniques. And I think that's an illustration of the title of the book, actually. It's called The Genome Odyssey. And I think that's a reflection of the fact that so many of our patients have these very long and eventful journeys. And that often they don't end for a long time. And we talk about trying to use the power of the genome to bring those medical odysseys to an end. I talk a little bit about Homer in the book, The Odyssey, and the kind of crazy 10 years that lead character Odysseus had as he's the king of Ithaca, getting back to his wife, who was living at home surrounded by suitors who were waiting for confirmation of his demise. And he's basically dealing with shipwrecks on one side, and he's dealing with siren songs and all sorts of other, giants with one eye who are trying to eat his crew, all sorts of things. And this is, these Greek poems are not called epic for nothing. But that name, Odyssey, came from Odysseus, the lead character in that epic poem. And it really, I think, describes well what our families go through. And I wanted, just before we maybe take some questions from the audience, to talk about another Odyssey as well that we ended up discussing in the book, and that brought us back together a few years later. And this was a little girl called Astria. And she first came to our attention when she was still in the womb, and she had a heart rate that was really too low as a little baby. Should be around 140 beats per minute, and hers was 60, 70, 80 beats per minute. So she came to Stanford Children's Hospital and was delivered by cesarean, and almost immediately came under the care of Anne Dubin, who's one of our pediatric electrocardiologists, because it became clear she had this genetic condition called long QT syndrome, where the resetting of the heart is just too long. And so we had actually set up recently a method of really leaning on collaborators. Steve Kingsmore had set up, he was at the time at Kansas, now at Rady Children's, had set up a method with Illumina of sequencing really fast. You know, that we talked about the price coming down, but it still would take a little while to get through the data and the analysis. And so we worked with the Illumina team, Tina Hambush from that lab, and managed to get the data back quite quickly. And we thought we found the answer, which was a sodium channel gene in the heart, which would allow us to really do what we call precision medicine and be able to give the right drug to the right patient at the right time. But a couple of our computer methods didn't agree with each other. And so we started scratching our heads as to how to deal with that, because it looked like we'd found a variant that looked so much like the smoking gun, the answer to this particular odyssey. And yet two of the computer methods didn't call it, and one did. So this became a new mystery. We leaned on the help of some friends at Personalis. This was a company that I describe how we started that in the book a few years on from when we dealt with your genome. We dealt with the genome of a family, the father of whom ended up being the CEO of this company. We started to look at genome analysis. And so I called them, and they rapidly swung into action and also sequenced the whole family. And what became clear at that time was that there seemed to be a chance that this little girl was one of those rare people who had more than one genome. And so that was the point at which we kind of decided we thought we need someone who can help us work out on a single cell basis what the genome of individual cells were. And that's not something that ever happens really in a clinical world, because it looked like she might have some cells with one genome and some cells with another one, but how do we ever go about proving that? And so I picked up the phone now to my friend from a few years back, and now I knew your number, so I got through. You remember that time? Tell me what.
I do. I remember getting the call, and my own research at that point had changed directions, and it had gone from trying to build the instruments to sequence the genome, and then it was clear that whole field was taking off, and big companies were going to be building these instruments, and the whole thing was on an amazing journey that there was not that much more to contribute anymore as a just university professor. And so attention turned to what would you use the machines for, and one of the things we became interested in was trying to sequence genomes from single cells. And it seemed like a very esoteric thing at the time, and I didn't think it would have a lot of practical application, but it let us dig into some really interesting scientific questions. And then so we had been building up that technology and kind of proving it out and getting it to work. And I had a terrific clinical fellow in my lab at that point, Chuck Gawad, who was kind of deep into that. And he was working closely with Norman Neff, who had been the same person who done the initial sequencing. And when we got the call from you about this, it was like, holy cow, you mean there's really an application for this stuff we've been doing? Because we certainly didn't expect it. And you guys had kind of had the right hypothesis, and you kind of clued into what you thought the problem was, but you couldn't prove it with conventional approaches. And this single cell approach allowed us sort of to work together to prove that your intuition was right, and that this young girl was, the technical word is mosaic, but had multiple genomes. And in a very unfortunate way, the genome in her heart was different from that in her blood.
Yeah, just incredible. And I remember at the time that he was a postdoc in my lab, but James Priest, who was a pediatric cardiologist, and he got together with Chuck, and really the two of them together, just really amazing work going after this to prove it. Because I think that the genome sequencing of all the tissue or all the blood was able to give us a clue. There seemed to be two genomes, but it couldn't really prove it. For that, we had to be able to isolate individual cells and then look at those genomes individually. And so it was just really fun and amazing that we were able to work together again and collaborate on that, and that you'd been working on exactly the technology that could help this little girl. And of course, what it showed, I think you mentioned, was that she was so-called mosaic. She had two genomes, and I was happy as part of writing the story up for the book to check back in with her mom and check back in with her, and she actually had a heart transplant later because of this condition, which made her a very unusual situation of being someone who is a mosaic, we call it, but there's also people who have not just two genomes that are derived from the same person, in her case, but sometimes a genome from another person, and that is sometimes called chimera. More Greek tragedy references here. And so she was actually someone who had a new heart with its own genome from another individual eventually. And prior to that happening, we'd actually taken even further the variant that was then proven by your studies to be mosaic in this little individual, and some colleagues at Gilead, a company, had modeled the exact change in the salt molecules that sort of go in and out the sodium channel. And then we passed over to this was just mind-blowing to me. We said, well, how do we test this? We could either try to create a model in the lab, or we could do a computer model. And so we reached out to Natalia Trianova, a Johns Hopkins bioengineer, really famous, probably the best known and most prolific computer modeler of the heart, and said to her team, I know that this might sound a bit crazy, but those computer models of hearts, have you ever thought of modeling a mosaic heart? And could we give you the information from these studies in terms of how mosaic, what percentage of cells, and also the sodium, the actual sodium currents? And they were amazing. Like everyone else for this little one, they jumped to the table, and suddenly there was another team there. And they basically built a whole new model to essentially model little Astria's heart and reproduce the presentation, which was the most incredible thing. It didn't happen first. There were a few twists and turns. I talk about the story in the book, but by the end, there was this living model of a heart in the computer. We could watch it essentially contract, watch the electrical activity kind of wash over it. So the fact that in the end she's done so well, and I saw a photo of her recently, we're just really happy about that. And the fact that we were able to bring so many teams together, that's been the incredible thing in these genome adventures, that just every time we call, people show up and bring their tools and technology, and it's just been incredibly exciting.
Yeah, I'm so glad to hear that she's well and healthy and that the transplant worked out. That's marvelous.
Yeah. Well, I know that we're about 2/3 of the way through our time. Evan, do you have some questions? We'd love to answer them if you do.
We do. Thank you. This has been a really great conversation, and what an incredible odyssey, as it were. Thank you both for sharing. And to those of you who are tuned in, thank you for your questions. It's not too late to ask if you're still holding on to one. Just drop them in the chat. The first question we have is from Gowrie, who is wondering, are you now encouraging all patients with a diagnosis or family history to get a genome sequencing? Is your information on genetic markers now fairly representative of all ethnic groups in the US? And how about worldwide?
All right, those are great questions. Maybe I'll do the first part, and Steve could talk about sequencing broadly and diversity. Sure. It's actually an ideal timing, I guess. Good, not planned this way, but timing with the book coming out, and also great timing from Gary, the questioner. Just 2 weeks ago, actually, we launched at Stanford. So this is 10 years after we talked about this first genome of Steve's that we worked on. Sometimes things take 10 years, but we actually launched the genome, we call it the backbone. So we've continued over the course of 10 years to be doing these panels where we look at a certain number of genes. The number has gone up over the years, but we still tended to look at for heart disease, for example, inherited heart disease, we've tended to focus on a fairly narrow number of genes. But we're now able to look across the whole genome much more powerfully than before, sometimes to look at millions of spots where there are variants that have a very small effect, but that add up overall to be a larger effect. We sometimes call those polygenic risk scores. But also we're able to identify many more genes. And so the idea, especially with genome sequencing coming down in cost, is that even if we only want to look at 5, 10, 50 genes, it now makes sense in the background to sequence the whole genome. Because then if we want to look at another 10 genes, instead of you having to have a new blood sample and a new sequencing, we just look up your file because the genes have already been sequenced. We just sort of open the next door and look down that way. And as I say, it can also mean that we can look across the whole genome. So that actually just got launched at Stanford in the last couple of weeks. And I think that this will increasingly happen. I think we might be the first to do the Genome Backbone for this testing, but I think many others are following along. And I think that that is something that is more broadly available. And it's partly because of this question of diversity that we're able to do that. But I'll maybe let Steve talk to that.
Yeah, look, the field has been criticized, and I think rightly so, for a lack of equity and diversity in the genome sequence. And you look in kind of the collection of all human genomes, and Caucasians are disproportionately represented by a lot. And that's not a good thing for the field. And, you know, I'm hoping over time it's going to even out. You know, it doesn't make sense from a lot of perspectives. One of which is that genetic diversity in Africa far exceeds the rest of the world. I mean, the cradle of humanity, and the diversity there is just like significantly bigger than the rest of the world put together. And, you know, what's made me feel pretty good recently is that, you know, there's been quite a bit of effort, not just to sequence African genomes, but to have African scientists lead that process. And there's a couple of genome centers in Africa right now that are, you know, really working on that question of genetic diversity across the continent, and therefore filling in our picture of genetic diversity across the human race in a way that's been missing. That makes me feel like, you know, the scales are starting to even out a little bit, and some much needed balance is starting to happen.
Yeah, and it's so important because every time we get a new result back, as I mentioned, the variance being like the needles in the stacks of needles, and the only way we can sort them out is by knowing what's common and what's not, and we need to have the breadth of ethnic diversity to be able to sort out the needles that really matter, that could be causing disease, from those that are actually common in the population. So yeah, fortunately we're in a much better position, and hopefully continue to move that further along.
Thank you both, and thank you, Gary, for that question. The next one comes from Susie, who is wondering, would you recommend utilizing this to better treat those diagnosed with autoimmune diseases? Thinking particularly of, I'm going to mispronounce this, Sjogren's syndrome, Sjogren syndrome, which presents differently with each patient. Some have neurological symptoms, some with autonomic, some with rheumatological, all with different treatment medicines with varying results. It seems like this would provide a better way to tailor both diagnosis and treatment as opposed to hit and miss. But what are your thoughts?
Well, I'm going to let Steve start with that one, because he is a world expert on autoimmune diseases.
Well, I wouldn't say that, but I have dabbled for sure. And, you know, the genome is interesting. What's also interesting is measuring the immune system. And so it's something that I think is also an important takeaway from the conversation tonight, is these sequencers are useful not just for sequencing the genome, but also for looking at the phenotype, for characterizing what's the current state of the person, the tissue, the organ, by sequencing the RNA that's there. The RNA are the messages that are made from the genome. And in particular, in the immune system, by sequencing the RNA in the immune cells themselves and looking at the repertoire of different immune cells, my hope is, and there's been quite a bit of excellent academic literature on this, that eventually that's going to translate into better diagnostics and be a good companion to the understanding the genome mutations that are also important for disease.
Yeah, and we haven't talked too much today because we've obviously wanted to chat through the stories that we shared, but the book has a lot of stories, little vignettes of patients with mysterious diseases. And what we've found is that the genome can be really powerful in helping solve those. And we talk about a number of kids with neurological developmental challenges, and the power of the genome to bring an answer to that. The other category of patients who often have medical mysteries are those who appear to have immune and autoimmune, sometimes even auto-inflammatory, the other part of the immune system, the inflammation. When you cut your skin and it gets red, that's inflammation. And sometimes that can get turned on when it shouldn't be. Just like in an autoimmune disease, your immune system can kind of turn on itself. And we've certainly seen patients in the context of the Undiagnosed Diseases Network, which is that group of doctors from around the country, medical detectives trying to solve the hardest cases in medicine. And we've certainly seen some unusual presentations of Sjogren's syndrome, in particular within that network. So I think that, yeah, I'm really excited about the next 10 years for our ability to use these kinds of genome sequencing tools to really look at the immune system in a way that we haven't been able to yet. And so, yeah, I hope so.
Beautiful. Thank you both. And thank you, Susie, for your question. The next one comes from Jamie or Jaime, who's wondering if you could talk about sequencing viruses.
Of course. And I think it's, so the very end of the book, because that was the part I was writing, in the last year. Of course, I do talk a bit about sequencing viruses, because of course that's at the front of everyone's mind. And I think we can draw a line from the sort of things we've been discussing tonight in sequencing the human genome and the technology that was really pushed forward so fast to try to really understand this massive genome of 6 billion base pairs, to the point where we're now, we're facing the pandemic. And if we go back, let's go back a year, just over a year, where the scientists in China sequenced SARS-CoV-2 for the first time, 30,000 bases of RNA, the messenger closely related to the DNA. Well, within a few hours of that happening, and certainly within a few days, there were already diagnostic tests that could be designed based on that sequence. So it's been an incredible, essentially an incredible story for science and the science of sequencing. And as Steve will talk a little bit more about the sequencing that he's doing through the Chan Zuckerberg Biohub here in the Bay Area, but also what it allowed us to do, and say us, but this is not from our work, but from many groups around the world, is to essentially to create a whole new era of genomic vaccines. Because if you have the sequence of the virus, you can use it to design a vaccine. And you may have read some stories in the last few months of those folks from Moderna or BioNTech or AstraZeneca, the Oxford team, NIH folks working with Moderna, to use that sequence to design genomic vaccines. And the leading vaccines that we have today, which is to say those from Pfizer and Moderna and AstraZeneca and J&J, they're all genomic vaccines. They're basically designed because we were able to sequence the virus. And they've been really spectacularly effective beyond, I think, our wildest dreams. So I think sequencing and genome sequencing, not of a human, but of a virus, has been such an important part of our response to the pandemic. And it's, of course, it's not over. The vaccines that have this tremendous efficacy, but we're starting to hear now about variants and how we need to follow those and make sure that our vaccines are responsive to those new variants. And that requires population level sequencing of viruses. And that is one of the things that the Biohub that Steve leads actually performs. So I'll let him chat a little bit about that.
Yeah, absolutely. So as Euan was saying, the only way to really understand when new variants are arriving, COVID being kind of the one in everyone's mind right now, is to be sequencing viral genomes as sick people are coming to hospitals and getting diagnosed. And the commonly used diagnostic tests for COVID, for example, and for many other viral infections, don't tell you to sequence. They just tell you, do you have that virus or not? And so you have to add on this other level of genomic surveillance to try to actually sequence those genomes. And that'll let you understand not just that you have that virus infection, but which variant you have. And it'll let you discover new variants as they come online. And the UK, United Kingdom, was very committed to doing this. And it's not surprising that the so-called UK variant was discovered there first. They were the ones who were looking the most. Because they were doing this as a matter of course. And so that kind of raised everyone else's kind of attention and kind of emphasized the need to be doing this at a much larger scale than had been done. At the Biohub, we've been doing some of that for the departments of public health here in California and for Stanford and UCSF. So we've been doing our small bit to contribute. We'll see where that goes. I mean, as Euan is saying, when you understand what the variants are that are sweeping through the population, you can then turn around and very quickly use that information to design more effective vaccines. Or you can test the existing vaccines against those variants to see if they're effective or not. And that's exactly what's going on right now in the medical community. Sequencing can also be used to discover, sort of allow doctors to diagnose infectious disease in a way that doesn't require them to have a guess ahead of time. Kind of the conventional way to do infectious disease diagnostics has been sick patient comes in, doctor asks for symptoms, makes a guess about what it could be, orders specific tests for those infectious agents. If they come back negative, you order more tests, and you get in this long cycle of trying to track things down. Sequencing allows you to do all that in one step and to do hypothesis-free infectious disease diagnostic. That's something my group has been interested in, others as well around the world. And there's now a number of clinical tests on the market that let you take that approach, where the doctor can come in, basically ask, test for all infectious diseases in one go, broadly speaking. And we'll see that, I think, more and more broadly used in medicine over the next several years.
Yeah, and I think that just to say again on the pandemic, it's been an amazing collaboration between all sorts of groups and coming together, of course, in the Bay Area, Stanford, UCSF, Berkeley, and the BioHub. We've been working with Ben Pinsky's lab at Stanford and the virology lab to do some sequencing for the research side. And we're part of a big international group that is sequencing genomes of the humans who are infected. So it's a really interesting situation where you want to sequence the virus. You also want to sequence the human, because we know that some humans have no symptoms and brush off the virus as if it almost wasn't there, and others are languishing in our intensive care units. And so we also are looking to see if there's a way we can understand who's most at risk. And so there's a few different ways that the sequencing can be used.
Thank you both, and thank you for that question, Jamie. We have a few more questions. The first one I want to ask is from Gretchen. Gretchen is wondering, I still do not quite understand having two genomes in one body, and wondering if you could explain that a little bit. I'm curious as well.
Yeah, absolutely. And we sort of use that as a bit of a shorthand, in that it's sort of the easiest way to describe it. But if you think about the time when a human is first made, it starts, we all start as a ball of cells. And if you think about that little ball of cells after the egg is fertilized, it becomes an embryo. It's kind of doubles, starts as one cell, then two, then four, then 16. And it's at the beginning, it doesn't look anything like a human. It's just a ball of cells. But one of the things to remember is that every time a cell divides, and this happens in our bodies as we grow up, some cells don't divide well, like in our brain and our heart, but many cells, we think of hair and nails and skin, those cells divide. And every time a cell divides, it has to make a copy of the DNA. Now, thankfully, our DNA, the copier, the enzyme that makes the copy of the DNA, is extremely good. It's just like an awesome proofreader. I've understood how important editors are as part of my amazing publishing team for this book. And one part of the editing team, big shout out to them, but one part of it is the proofreaders who go through at the end once the content is set and the story is set, and they make sure that you didn't miss anything. And actually our cells have that as well. And DNA proofreaders are extremely good. I mean, we were just talking about the virus. Interestingly, the RNA virus, coronavirus, isn't that good at proofreading, which is where the variants come from. And so that is, especially if we let the virus run amok in populations. But DNA is actually very good at proofreading. So most of the time it gets it exactly right with really low error rate. But every now and again, it does make an error, and an error that isn't corrected. So you imagine that you're now a human at the very earliest stage, just a few days old, and you're 64 cells. And as you divide to 128 cells, one of those divisions creates a variant. Now you have 3 billion positions in your genome where that variant could be, and obviously most of them, it's quite likely it wouldn't have a major effect. But what if that one error happens within a gene that's really important, let's say a channel for your heart? Well, then now you end up with one cell, in this case of that example out of 128, actually our patient, it happened a little earlier, where it was about 10 cells out of 100, which has that one extra variant. It's not that the whole genome is different. It's actually only different, as far as we know, at that one spot. But just by play of chance, that one spot in little Astrea was really important. So that's basically how it happens. And then as they grow up, those cells divide, and that group of cells goes on so that they end up with eventually only 10% of their cells with this one variant in that gene, and then 90% with the original genome. And so we ended up modeling that as well. We don't know where the cells are exactly. For example, in the heart, the Natalia's team modeled could be clumps of cells with the different genome, or it could be like almost like a salt and pepper. And in the end, that was one of the things we could do with the computer model that was impossible any other way.
Fascinating. Thank you. That was a clear explanation. I think even I got that. Thank you. Thank you. I have another question from Susie, which is, can you, wondering about privacy in a for-profit healthcare world. If you get your genome sequenced, what measures are in place to prevent insurance companies from accessing and using this information to determine who to cover or who not to cover?
Yeah, good question. Do you want to start with that?
I can take a whack at that. So I have a story to go with it too. But just before the story, so under the first President Bush, he signed an act into law called the Genetic Non-Discrimination Act, GINA. I'm missing a letter there, but and so it is illegal in the U.S. to discriminate against people based on their genomic information, unless they're members of the armed forces. There's a few exceptions where it can be done, but broadly speaking, you're not supposed to be discriminated against. So there is the protection of a law. Whether it's ever been enforced, I have no idea. And you know, when I sequenced my genome, I published it. And so I gave it all away for everyone to look at, knowing that I was, A, had the benefit of protection of this law, but B, knowing the law had never been enforced and not knowing if it would be held up or what would happen. So it felt like taking a little bit of a risk, but, you know, one that I thought was worth it for science. And the story I'll tell you is that, you know, I think that's, you know, missing the forest for the trees to be worried about genetic discrimination, because insurance companies can discriminate you based on everything else about you anyways. And I remember at one point, my family, who had been telling me go get a cardiologist, also said, you should get more life insurance. And I at one point gave in and called up a life insurance company and said, you know, all right, what do I need to get more life insurance for I had young kids, what happens, something happens to me? And they said, no problem, we'll write you a policy for however much you want, but you need to pass a physical exam. And we're gonna go measure your heart rate, your pulse, we'll take the echocardiogram, we'll measure your phenotype, everything about you, we'll take a blood test. And if there's anything we don't like about that, we're not gonna write you an insurance policy. So they're already able to discriminate against you based on every other property, never mind your genome. And I think that's the larger issue that everyone should be concerned about in this country.
Yeah, it's a really well made point. And, you know, the Affordable Care Act had some protections for pre-existing conditions that people are probably used to discussing. Gina, you mentioned from the earlier, has certain protections for sure. And you mentioned a couple of the exceptions. One of them, and it's very relevant for your story, is actually life insurance. So they can't discriminate on the basis of for employment or health insurance, but they can discriminate on the basis of life insurance. So we often recommend our patients, if they are thinking of life insurance and are also thinking of genetic testing, to do it that way. I mean, as you say, I think our world needs a few more corrections to take away that discrimination just much more broadly, but genetic discrimination is absolutely part of it. And genetic information is a bit different in the sense that it can be a very specific signature for an individual, albeit one that's quite complicated, and matching that with an individual is also quite complicated.
Yeah.
Makes sense. Thank you. Thank you both. And I think that's all our questions for the evening. Thank you all who have joined us for this conversation. It's been wonderful and an honor to have you both here. Thank you for your important work. And Dr. Ashley, congratulations on the book.
Thank you so much. And thank you for having us. Good luck with Booksmith. I'm so happy to be part of it. And thank you so much for hosting tonight. Steve, thanks a lot for joining us.
Thanks for being here, Steven. This has been a great talk, everybody. The Genome Odyssey, the link is just below the video embed. Click through, get it for yourself, get it for your friends, and get it from Booksmith. Thank you all, and take care. Hope to see you in person soonish. Until then, please take care and be safe.
That'd be great. Take care, all.
Good night, everyone.